Hi, and welcome back to the MicroMolar podcast. This is our health and wellness series brought to you in collaboration with the Maragin Health Foundation.
Today, we're diving into a topic that sounds like it's been pulled straight from a science fiction novel, but has very real roots in biology: parabiosis.
It really does. At its core, parabiosis is an experimental technique where two living animals, most often mice, are surgically joined together so they share a single common circulatory system.
Right, so they're literally sharing blood. And the most fascinating version of this is called heterochronic parabiosis. That's where scientists pair an old mouse with a young mouse.
And this is where things get incredible. The old mouse often starts showing dramatic signs of rejuvenation. We're talking about improved muscle repair, better cognitive performance, and enhanced tissue regeneration across its body.
Wow. So it's like a fountain of youth, but there's a flip side, isn't there? The young mouse that's paired with the old one can actually start showing signs of premature aging.
Exactly. It's a two-way street, which tells us something profound. There are circulating factors in blood that can strongly influence the aging process, for better or for worse. So how does this actually work?
Well, the shared circulation allows for an exchange of pretty much everything in the blood. We're talking proteins, hormones, and other metabolites. Think of them as molecular signals or messages being passed from one animal to the other.
Mhm. And while cells, like immune cells or even stem cells, can also cross over, the scientific consensus is that the main effects are driven by these signaling molecules, not by the cells themselves taking up permanent residence.
That makes sense. So, this obviously leads to the big question: if this works in mice, what about in humans? Has this been
It
tried?
has, but not by surgically joining people, of course. The research motivated clinical trials involving infusions of young plasma into older individuals, particularly for conditions like Alzheimer's disease.
And the results?
They've been mixed, unfortunately. Early studies were small, and so far, there's no definitive clinical evidence of a broad benefit. Plus, it brings up major safety and ethical concerns, and regulators have even issued warnings against unproven commercial uses.
Right. So, the focus is shifting. Instead of using whole plasma, the goal now is to identify the specific molecules, the good factors in young blood and the bad factors in old blood that are responsible for these effects.
Precisely. If we can pinpoint those molecules, we might be able to develop targeted drugs or other therapies that can deliver the benefits without the risks of a full blood plasma transfusion. It's a much safer and more precise approach.
So, to sum it all up, the strange experiment of parabiosis has shown us that aging isn't just a process locked inside our cells. It's heavily influenced by systemic factors circulating in our blood.
And while the idea of young blood transfusions is dramatic, the real, promising future of this research lies in identifying the specific molecular signals and creating evidence-based therapies that are tested in rigorous human trials.
A fascinating look at the cutting edge of aging research. A big thank you to the Maragin Health Foundation for their collaboration on this series. And thank you all for listening.
Join us next time on the Micromolar Podcast.